Hello, wonderful AP Bio students, and welcome to our video on photosynthesis details. Now, this might look kind of confusing, but we are going to break it down into two reactions, the two stages of photosynthesis. We have the light reaction occurring here, and this occurs in the thylakoid membrane, and that light reaction produces energy needed to get the Calvin cycle going, which is going to give us our carbohydrates, which are then broken down for energy by things like me and you. So let's dive in. Now, when we look at the light-dependent reaction, we know that it's going to happen in the thylakoid membrane. That's where all like the mechanisms are to have this reaction work. And let's look at some of the inputs. So we're going to input into this some water into the light dependent reaction. And we're also going to put in some light energy. And also really important to remember that the light dependent reaction needs is NADP+, which is an electron carrier, and also a DP plus PI. Okay, so those are all the ingredients we need for this reaction to work. And what comes out, of course, NADP+ is going to be reduced by a hydrogen ion and NADPH. We're also going to get some ATP. And our waste product is going to be oxygen. So when we look at the Calvin cycle, these two things right here, ATP and NADPH, are going to go in the Calvin cycle. And the Calvin cycle occurs in the stroma of a chloroplast. And the Calvin cycle needs ATP, NADPH, as well as it also, let me do some blue here, it also needs CO2 and an important enzyme called rubisco. And these are going to give us these are going to give us our ADP plus PI and our NADP plus. And it's also going to give us carbohydrates. So it's going to give us glucose. So let's get into the details of these. First, we're going to look at the light-dependent reaction. And I would like you to draw this in your notes. This is the membrane of the thylakoid right here. So if this is the membrane of the thylakoid, this is going to be outside of the thylakoid. So over here is going to be the stroma. And here is going to be the thylakoid lumen. So remember, we imagine the thylakoids as kind of like deflated flat balloons. We're going to do a process that's going to fill them up a little bit. Now, before we start that process, we need to understand our key players. We have a protein right here that's actually kind of serves as like a reaction center. It receives the light. And that protein is called photosystem. I'm just putting PS, but you should write photosystem. Photosystem 2. And this is where chlorophyll hangs out. So the chlorophyll in photosystem 2 is actually what absorbs light. And we also have another photosystem which does the same thing, called photosystem 1. And the only reason that 2 is first is, that 2 comes before 1 is that actually, photosystem 1 was discovered first, so it was named photosystem 1. And once they discovered the second photosystem, they just called it photosystem 2. So it's a little out of order and weird, but that's just how it is. We're also going to revisit our wonderful friend, which often looks kind of like this, called ATP synthase. And within the membrane, we're also going to have some proteins. You don't need to know the name of them, but they are involved in the process. It's actually one that's kind of shaped like that. Some proteins, integral proteins that are involved in this reaction. So let's start off. So we're starting off first at photosystem 1. And at photosystem 1, we need light. Now light hits photosystem 2. Light hits photosystem 2, and the chlorophyll within photosystem 2 absorbs the light and kind of concentrates it. And the whole reason it's trying to concentrate it is to break up a molecule of water. And a few things happen when we break up a molecule of water. The first thing that happens is we have some waste. Our waste is going to be oxygen. It's actually half of molecular oxygen for every molecule, right? Because we only have one oxygen here. So we need to break 2 water molecules to get molecular oxygen. So technically, when we break one water molecule, we get half of our O2. And that's our waste product, right? That's what plants essentially breathe out. So we have light, and it's concentrated, and it's breaking apart water, and that gives us some really awesome things. That gives us some high energy electrons, and it also gives us some hydrogen. Okay. So photosystem 2, we have light concentrating energy that's being absorbed by chlorophyll. It's breaking apart water, and we have excited electrons, and we also have these hydrogen protons. And these hydrogen protons, you're probably going to guess, are going to end up being pumped into the thylakoid lumen. And the thing that's going to pump them through is the energy of this electron, which is going to act as the energy needed for these integral proteins to physically pump these hydrogen ions into the thylakoid membrane. So we have hydrogen. Of course, this hydrogen came from our water, right, that we just broke apart. So the energy of the electrons is used to excite these proteins, which pump the hydrogen ions into the thylakoid lumen. So the thylakoid lumen actually ends up kind of filling up with these protons, much like a balloon, right? So then we want to make this process really efficient. So we're going to do that whole thing again, except for we're just going to use light energy. And light energy is going to hit photosystem 1, and it's going to excite this electron a little bit more to be able to pump more hydrogen ions into the thylakoid lumen. And when this electron is at the end of its road, it's all worn out, it's no longer excited anymore by light, it's picked up by our famous electron carriers, which what in this case is NADP plus, picks up the electron and becomes NADPH. All right, so the final electron acceptor at the end of we have 2 electron transport chains here. Instead of oxygen, like in respiration, we have a molecule called NADP plus that picks it up, and along with a hydrogen, right, becomes NADPH. So now that we have this high concentration of hydrogen protons within the thylakoid lumen, filling it up like a balloon, right? The ATP synthase takes advantage of this concentration and actually funnels the hydrogen through the ATP synthase. So the hydrogen is just flowing through, it's following the concentration gradient. Now ATP synthase isn't considered an active protein. This isn't considered active transport. This is passive transport. It's fueled by the concentration gradient of these hydrogen ions. And the ATP synthase is shaped really specifically so that when these hydrogen ions flow through it, it actually spins. And it spins really, 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 really fast, like unimaginably fast. And the spinning action of that actually mechanically binds ADP plus PI to make ATP. Okay, so that is our light reaction details. Let me go over that one more time. We have photosystem 2, which receives light energy and water as well, and that light energy is concentrated and breaks apart water. We have our waste product, which is oxygen, and from that water breaking apart we get these high energy electrons, and we also get hydrogen protons. Now these high energy electrons are used to excite, basically energize these proteins, which are going to be our electron transport chain. I can add that right here. Here's the ETC, the electron transport chain, which is going to actively pump hydrogen protons into the thylakoid lumen, filling up the thylakoid kind of like a balloon with hydrogen protons. And then we look over at photosystem 1, where this electron is gaining a little bit more energy from light photons, right? And is then fueling another electron transport chain to pump more hydrogen ions into the thylakoid lumen. Now that electron, that tired little electron at the end of the electron transport chain, needs to be collected, right? It's like the mom waiting at the bottom of the slide for her kid. The kid needs to be collected at the end so the rest of the kids don't pile up on the slide. So we have NADP plus, along with a hydrogen proton, collecting that kind of worn out electron and becoming NADPH. This NADPH is then going to go to the Calvin cycle, which we will talk about. So now we have this high concentration of hydrogen protons, and hydrogen protons get funneled through ATP synthase, and the act of it funneling through actually physically spins ATP synthase and mechanically binds ADP, an inorganic phosphate, to ATP. Now here's a quick little drawing of that. That's actually quite a bit better than my drawing. They use the word photons for light, so light travels in kind of little packets of energy called photons. So the photons and water hit the photosystems. We have our electron transport chain right here. We have NADP plus an hydrogen proton, accepting that final electron right there and becoming NADPH. This goes, of course, to the Calvin cycle. Just say it goes to Calvin, goes to visit Calvin. And then this is a little bit flipped from our picture. I feel like the way that we did it, it works just a little bit better. And these thylakoid, the hydrogen protons build up within the thylakoid interior space and flow through that ATP synthase, making ADP plus PI into ATP. So there we have it. Now let's find out where this ATP and this NADPH, what's its whole role in the Calvin cycle. So the Calvin cycle is named after none other than Melvin Calvin, and the Calvin cycle is often mistakenly called the dark reaction just because it doesn't require light to occur. However, it's actually the sugar-making reaction of photosynthesis. So in the light reaction, we created NADPH and ATP from water and light, and our waste was oxygen. And now these guys, along with CO2, are going to enter the Calvin cycle and make sugar, which is C6H12O6. So here we go. So let's first look at the players involved in the Calvin cycle. So you're going to need to draw this in your notes. We have, of course, this is where CO2 comes into play. This is what we breathe out. There's also going to be this important compound called ribulose biphosphate, and it's basically a carbon-based molecule that is phosphorylated. And then we also have this important enzyme. This is an enzyme right here. And this enzyme is called ribulose biphosphate carboxylase. So that's a fun word to remember. And its name kind of describes exactly what it does, kind of like all enzymes do. ATP synthase, it makes ATP. So this actually combines carbon dioxide and ribulose biphosphate. So it's called ribulose biphosphate carboxylase. And we also are revisiting our friend PGAL, which is phosphoglyceraldehyde, but a lot of times it's called G3P when it's talked about in the Calvin cycle. So let's get it started in here. Let's start with blue because I just feel like blue is my color today. So we are going to start with CO2. And I'm actually going to put numbers to these because at the end I'm going to have you count the carbons and double check my math for me. So we're going to start with CO2, and CO2 enters the carbon cycle, and it's a cycle, right? So it's going to go through this cycle and it's going to end up as that something. And that something that it ends up is ribulose biphosphate, which is RuBP. And RuBP is a 5-carbon, 5-carbon molecule with 2 phosphates, biphosphate, ribulose biphosphate, that are attached to it. Now, the thing that actually puts together RuBP and carbon dioxide is, you probably guessed it, ribulose biphosphate carboxylase. Our enzyme, our superstar enzyme here, is going to be the man. Actually, this enzyme is the most, it's the most common protein on Earth. So it's a protein that's integral to this process, and it's the most common protein. It's an enzyme on Earth. And so Rubisco combines carbon dioxide and RuBP, and this process is a lot of times called the carbon fixation. And they together, where is my blue? There it is. Why is blue so hard to find? They together form 12 pGals, or G3Ps. Now, if you remember from learning glycolysis that pGals are just a 3-carbon molecule with a friendly phosphate group attached to the end of it. So that's phosphoglyceraldehyde, or glycerol 3-phosphate is G3P. So, and during this reaction needs a little help. And it needs a little help from some of our products from the light reaction. And those products are, you probably guessed it, are ATP and NADPH. So the energy from these 2 molecules is used to kind of help this reaction along. And there's actually several intermediate steps here. Help this reaction along, and then they get used up. So ATP and organic phosphate, and also NADP+, and they're headed back to the light reaction cycle. So now we have our 12 pGal. And our 12 pGal, well, some of them continue to go through the cycle, and some of them leave the cycle, and there's actually a number to that. So about 10 pGal keep going to become RuBP, and the math works in terms of the carbons. We have 30, oops, sorry, I didn't put my number here. So we actually have 6 RuBPs, and so if we have 6 5-carbon molecules, that's 30, and then we have 10 3-carbon molecules, that's 30 carbons as well. So our carbon count is keep in check here. Now, where did those other 2 pGals go? Where those 2 pGals are going to be the products of our process here, and those 2 pGals. So if we look at pGals, they have 3 carbons. Those 2 pGals become a 6-carbon molecule, and the most common one it becomes is, let me put a little arrow here, scribble that out. Our most common one is C6H12O6. So it takes 2 pGals to make C6H12O6. And that's it. That's how we make glucose that is stored in plant cells and used to make the plant grow. So here is a much more complicated diagram of this. We know there's our input phase right here. This is where carbon dioxide comes, and Rubisco, the enzyme, binds it with RuBP, which is a 5-carbon phosphorylated molecule. And that RuBP goes through some reactions and becomes phosphoglyceraldehyde. And that G3P, glyceraldehyde 3-phosphate. That G3P, we have 10 of them that keep going to keep the cycle going, and we have 2 that leave. And when we get 2 of these, we get a glucose or other carbohydrate-like compound. So, in review, we know that photosynthesis is a 2-stage process. It occurs in chloroplasts of plant cells, specifically the thylakoid membrane, the thylakoid lumen, and the stroma. The light reaction uses chlorophyll to absorb light photons, and that chlorophyll is found in the photosystem 2 and photosystem 1. The whole purpose of the light reaction is to produce ATP and NADPH, which then goes, of course, there's my twisty arrow there, to the Calvin cycle. Photosystems, the electron transport chain, and ATP synthase are important key items found in the light reaction. And those things that are produced in the light reaction go to the Calvin cycle, and the Calvin cycle uses ATP and NADPH to produce storable energy called glucose. And that enzyme that's like the awesome enzyme of the world that is most common is our rubisco. That makes this whole thing happen. And that's photosynthesis. I hope you enjoyed. It's a lot to take in, so watch and rewatch, and also watch the detailed animation videos on this page. Thank you. Bye-bye.